G-protein coupled receptor. The G-protein coupled receptor is attached to the plasma membrane, with one side being in the cytoplasmic side and the other facing outside the cell. This is a G-protein. A G-protein is a protein that works with the G-protein coupled receptor. The blue Play-Doh, right here, represents the GDP, which is bonded to the inactive G-protein. The pink Play-Doh is the phosphate group, which bonds to the GDP to make it GTP. The orange Play-Doh represents a signaling molecule. The signaling molecule binds with the G-protein coupled receptor to start a reaction. The green Play-Doh represents an enzyme. This enzyme starts off inactive, but when it combines with the GTP, it becomes active.
When the signaling molecule binds to the extracellular side of the receptor is activated and changes shape. The cytoplasmic side of the G-protein receptor then binds to the inactive G-protein. By binding to the G-protein, a phosphate group is added to the GDP. This makes it GTP. This activates the protein.
The active G-protein dissociates from the receptor, moving along the cell membrane, and binds to the enzyme, altering its shape and causing a cellular response.
Changes in the enzyme and G-protein are only temporary. The signaling molecule is then removed. Then the active G-protein then hydrolyzes its bound GTP to GDP again, rendering it inactive. Because the G-protein is inactive again, it leaves the enzyme and returns to its original state, allowing it to be available for reuse. Because it also functions as a GTPase, which allows it to rapidly shut down and become inactive again when the signaling molecule is not present anymore.
One example of this process is epinephrine, which is a hormone released by the adrenal medulla upon stimulation by the nervous system in response to stress.